Yeah.
Okay, yeah. All right. So, welcome again after the short break, all of you. And we have the next speaker of the day. It is my privilege and pleasure to introduce Dr. Saikat Chowdhury. Dr. Saikat did his PhD in the Pennsylvania State University, followed by a postdoctoral fellowship at the Scripps Institute, California. After his postdoc fellowship, he joined the Department of Biochemistry and Cell Biology, Stony Brook University, New York, where he continues to hold an adjunct position. In 2021, he joined the Center for Cellular and Molecular Biology as a senior scientist. His lab research focuses on understanding how different cytoskeletal elements work in unison inside the cell. And he investigates the molecular basis of regulation of these networks and how these participate in the various cellular functions. For these, he uses multiple techniques, which include the cryo-electron microscopy and image processing methodologies, along with various biochemical and biophysical techniques. With this very brief introduction, I thank Dr. Saikat for joining us today in this national level online introductory workshop on Cryo-EM, which we believe will be the first of a series of workshops focused on enhancing the use of this cutting edge technique in the Indian research landscape. Dr. Saikat, please.
Yeah, thank you very much for the kind introduction and also for the invitation. So just let me get started with the presentation mode. So yeah, I mean, it's my pleasure to present in such a forum. So today I'm not going to talk much about my own research because 1 hour is not enough for talking about research as well as giving an introductory sort of a course on microscopy. So I'll briefly touch upon what I do, but primarily I'll focus on the tools and techniques and give an overview within 1 hour, whatever I can, crash course of something that has been developed over several decades, almost more than 50 years it has taken for Cryo-EM to come to where it is today. And I think you must have heard a lot about it from Dr. Natesh earlier. I have been doing different structural biology methodologies, and in the last 10 to 12 years, I've been primarily using Cryo-EM as a tool to understand different macromolecular structures and using it for my own research and stuff. So I'll give a overview about where things stand and how we use EM in general, why is it necessary and stuff. So this presentation has been designed keeping in mind the diverse audience that we have, and some of you may or may not be familiar with structural biology or even cryo-electron microscopy, let alone cryo-electron microscopy. But I'll try to explain as best as I can. And in between, if somebody has a question, maybe feel free to interrupt if possible. I'm not very familiar with Google Meet, but I'll try my best. Okay. So this is a children's book. It's a French book. It's called Le Petit Prince, The Little Prince. And I always find this statement in this particular book quite philosophical, though it's a children's book. What is essential is invisible to the eye. So in context of this book, it means like friendship, love, emotion, and all these things which are feelings in human being. That's kind of invisible to the eye. But in general, also things that are essential for life, the basic fundamental units of life, that's also kind of invisible to our naked eye. And we try to look into that. We don't invent per se most of the things that we do in our lab, but we try to essentially discover what Mother Nature has made and a scale which is like really tiny and almost invisible to our naked eye. So before I start my talk, just imagine all the biochemistry textbooks or cell biology textbooks that you have. And for a moment, think about all the covers and all the pictures that are there. So if I just black them out and just show you the title of the book, it's pretty boring. There's nothing much that you can basically guess about the book other than the title, who the authors may be. Many of them are pretty famous trailblazers in their own field and stuff. But then if I uncover this, then you'll see all these pretty pictures, which are primarily structures of these macromolecular complexes that do different kind of functions in our body and in life in general. So basically, I mean, it's important. I mean, people who do computational biology, people who do any kind of evolutionary biology, they know that actually the structure is what determines the function. And primarily, in order to maintain the functionality, a lot of times these structures or the amino acid building blocks of these proteins and stuff are highly conserved so that their function can be carried on from across different species, the common function that these particular class of proteins or macromolecules perform, right? So it's important for us in that context to understand how these things look like, we use different tools in general, like genetics or even biochemistry and molecular biology, to understand how these things function. But structure ultimately tells you what's the three-dimensional shape of the molecule, and why that shape or the juxtaposition of the different amino acids and building blocks impart its function, right? So it becomes important and interesting, rather than just the pretty aesthetic pictures that we see also on these biochemistry textbook and stuff. So I'm going to talk about how do we see these macromolecules. As many of you know, we are talking about structural biology, about Cryo-EM. So of course, that's how we see it. But why? So we have different structural entities or different if you look at biology across scales, starting from human beings to fruits to flies and ants, all of these most of us can see with our naked eye. But if you have poor vision like myself, then we use a kind of a spectacle or a glasses to correct for it, or wear corrective lenses to actually look into it or look at it. And then we can look into finer structures, like we can look into cells or bacteria, or even like chromosomes and stuff that can be seen by light microscopy. And then all the way, if we go down to subcellular components, and then finer and finer or smaller and smaller molecules, such as, say, organelles or even proteins or macromolecular complexes, for that we need something even more high-end technique or tools, such as electron microscopy, which sounds very fancy. But yes, we do need these fancy tools to look into at that realm. And then ultimately, if we look into subatomic particles, and you need even more complicated tools like the Large Hadron Colliders and other things that exist at CERN, or particle accelerators and stuff, where they smash atoms together and then they try to detect some of these subatomic particles and stuff. I mean, what's interesting is we are talking about a dimension scale which is really, really tiny, which we cannot see by small eyes. We need these tools and techniques to visualize them. And if we think about things which are like astronomically big, like, for example, all these galaxies and planets and stuff, you also need fancy tools, maybe even more expensive tools, to look at that larger dimension. So we are talking across different scales right now. Some of these are astronomical stuff, which we need fancy tools and telescopes to look at. And right across the scale, on a smaller realm, then we need all these techniques to actually see these smaller and smaller things. Now, what generally as biologists, or in my lab, what we try to understand is we want to look into how these macromolecular assemblies or these proteins are organized in context of a cell. But before we get into cell, even we purify them, and then look at what is their 3D ultrastructure, how they are organized, how does the structure eventually gives rise to its function. And then ultimately, if there are any kind of alteration in the structure, primarily due to mutations and stuff, now how that impairs the function of this particular protein, and how that can contribute to certain diseases and stuff. That's sort of the overarching goal for any of the biology labs that exist, that we try to understand how all these things kind of work together. And some of us look at this in context of structures, or try to understand the function from a structural perspective as to how these things are organized, right? So many of you have seen these nice basically drawings or paintings by David Goodsell. You can see them in many of the websites and stuff, including PDB and places like that. But these all come from different cell biology, as well as many structural biology techniques, primarily microscopy and crystallography, that have kind of given us all these structural information. But in context of cell, you see how crowded the cell is and how these different things are organized and stuff, right? So these things actually are the things that we gain from all these structural techniques and methodologies. Now, why do we need such fancy things? Now, if you think about generally just a light microscope, we can't see things which are really tiny in the level of some nanometers to angstrom level. That's primarily because visible light's at bigger wavelengths. Like, for example, somewhere between 400 to 700 nanometers is within which your visible spectra lies. So you cannot see things which are really tiny, smaller than the wavelength of these lights. So that's when we need something which have a comparable wavelength to the dimension scale that we are interested in looking at. So I won't go too much into this math and details. So what we do is we use electrons and we accelerate them at very high voltage, and that imparts a lot of energy. And now energy can be converted into wavelength. So we get a really, really tiny, in the angstrom level or even sub-angstrom level wavelengths we can achieve because of the high voltage across which we accelerate electron inside the column of an electron microscope. So what I'm going to talk about right now is essentially using electrons as waves and then trying to visualize these macromolecular assemblies and complexes that are present in our cell. So as I mentioned just now, so electrons, when we accelerate them at very high voltage, these are in picometer length dimension as compared to nanometer dimensions. So that's why we use electrons as a source of illumination for visualizing these structures and stuff. So this is a typical like a light microscope, which many of you have seen, or they are there in your, I mean, this is primarily directed to the students. They have seen this in their labs and stuff. So where you have a light source, then basically the beam of light is condensed, it's projected onto an object or a specimen, and then you have an objective lens that forms the image of an object. And then you have projector lenses, which basically, and then magnifying lenses, which essentially magnifies the image of the object that you want to visualize. And then you use glass lenses, which can bend light waves, visible light waves. And then you use that to basically direct the light and then magnify it to see the image. Now, in the case of an electron microscope, you have the same sort of lenses, but because it's electron beam, you essentially cannot use a glass lens to bend an electron beam. So based on the electromagnetic properties, what we do is we use something like an electromagnetic lens. And here's just one of those electromagnetic lenses, which looks completely different from what a glass lens would look like. So you have these same set of lenses. You have the condenser lens to condense the beam, projected onto a specimen. Then you get an objective lens, which creates the first image of an object, which is inverted. And then you have the projection and magnifying projector and magnifying lenses, which essentially magnifies it several thousands of times to basically look at hundreds of thousands of times to essentially look at the magnified image of the object that we are interested in seeing. So this is sort of a schematic, and then a typical electron microscope. Nowadays, it looks very different, but this is how it would look like. It's just an inverted version of your light microscope, where you have a source, which is an electron source or an electron gun that generates the electron beam, and then it goes through all these electromagnetic lenses and then falls on the specimen. You get a magnified image. So there are several kind of electron microscope. There's scanning electron microscope, which basically once the beam hits a sample, whatever is the backscattered electron from the sample, people try to get the surface information about the sample from SEM, or scanning electron microscopy. But the kind of microscopy that I'm going to talk about is called transmission electron microscopy. So essentially, when you put a specimen or a sample in the electron beam, the beam goes through the specimen. It is like if you have broken an arm or something, or you want to get a chest X-ray done, you go to a radiologist, and then you stand in front of an X-ray generator. The X-ray beam goes through your body, and then the radiologist essentially can see everything that is present inside the body on the X-ray film. The beam gets transmitted through the patient's body. It's the same kind of thing over here. The electron beam gets transmitted through the specimen that you want to image. So that's why we call it transmission electron microscopy, or TEM. Okay, so the kind of microscopy I'm going to describe right now that we have been extensively using for visualizing molecules, or even for structural biology in general, is transmission electron microscopy. This not only doesn't give the surface feature, but actually gives all the information that is present inside the object that we want to view. Now, many of you have seen these in your textbook, a general structure of a cell. These come from classic cell biology textbooks and stuff. And these are all kind of TEM images, or transmission electron microscopy images, where people have put heavy metals to stain certain like membranes and things like mitochondria and other organelles got stained by these heavy metals, which basically scatter a lot of electrons. And then you get a contrast in these otherwise non-contrasty biological specimen. And then you can see all the inner details of the cell. So this is typically what most of us are familiar with from the standpoint of a cell biology textbook. And this has given us treasure trove of information over decades, which has helped us understand how the cell looks like, how different kinds of cells look like, and what are the different organelles and stuff. And this has sort of revolutionized cell biology in general over several decades of time span, and which has given us a lot of information about how things look like inside the cell. Now, this again comes from transmission electron microscopy, because here you have a section of a cell. The electron beam has gone through these, and whatever has scattered, and that has given rise to this nice micrograph, or an image of a cell, where you can see all these finer details. Now, the capability of an electron microscope is that you can even see atoms. So this is usually what material science people would do. So these are all these kind of oxide materials, either ruthenium oxide or strontium, and then titanium oxide. And you see these little dots over here, and these dots all correspond to individual atoms. So you get a resolution of 2, or even better, like close to 1 angstrom resolution, essentially, where you can see the individual atoms that are present within these samples. But unfortunately, with biological specimen, we are almost near that resolution realm of seeing individual atoms, but we can't. And why is it so? That why we can't see these individual atoms that make up our biological molecules? What we get is near atomic, that is as close to an atom as possible that we can see. We get an experimentally derived map into which we build an atomic model, which is essentially like the atomic representation of the experimentally derived map, which is then we deposit into protein data bank. which many of you may have downloaded to look into these structures and stuff of these complexes. So unfortunately, we are not there yet, but we are getting there. And why is it so that we are not there yet? So first and foremost, most of us are made up of mostly water. So there's almost 80% water is the content of any cell or any living organism. So when you put it in a vacuum, and if you do that at a room temperature, it'll all just evaporate. It'll all be gone, and then it'll be just a shriveled, dry mass of protein. So we are essentially going to destroy the sample. So what we need is we need to preserve the ultrastructure as best as we can, so that we can image these in a vacuum. The reason we have a vacuum inside the column of a microscope is because electrons also behave like particles, and they can get scattered by the air or any other atoms that are floating around from the gaseous air mixture if there wouldn't have been any vacuum. To prevent any of these scattering phenomena and just to let the beam hit the sample without getting scattered in between is what we want, and that's why we have a very high vacuum inside the microscope. And in this vacuum, if you put any of these samples, it'll just basically at room temperature, everything will just boil off or evaporate. That's essentially what happens if an astronaut doesn't wear a spacesuit and goes outside and does a spacewalk. It's complete vacuum, and the body temperature, everything will just boil off. The entire blood will go away. So that's why they need that spacesuit to put some pressure and keep the astronaut protected in space. In addition to that, at the high voltage and the high energy at which we image these objects, I just showed some mathematical equations where we convert the energy that is possessed by the electron into a wavelength to basically use it as a wave. Now, at that particular energy, at that much amount of energy, it's almost like having a nuclear, a bomb or an atomic bomb just exploding at small, minuscule level. So we'll just literally fry up our sample. So we need to preserve the sample as best as we can, and constantly as we are imaging, the sample is undergoing radiation damage. That is also kind of true for X-ray, because these are high energy electromagnetic waves, which also generate certain free radicals, which can break bonds and stuff as we collect data in X-ray. Same goes for EM. Because of the high energy of the electrons, it's constantly breaking bonds and stuff, and then that can destroy the biological. It actually does destroy the biological specimen as we are imaging, but there are ways today we can actually capture as best as possible without destroying the sample and maintaining the integrity of the sample. We can collect these data and get really a treasure trove of information from those data that we are collecting nowadays. So one way that many of us are familiar with is essentially embedding the specimen in something like a heavy metal, like a stain, that sort of kind of preserves the ultrastructure as best as possible. It's called as negative staining, because essentially the sample is not stained, but you have the surrounding which gets stained, and wherever the sample is occupying in that stain, that region is not at all stained. So it's essentially the background which is stained and not the biological sample. And so essentially you take your protein, you put it on a sheet of carbon or a thin sheet of carbon, and then basically apply a heavy metal stain on it, and you get a casting of the stain. And because of the high density of these stain, they scatter a lot of electron, which leads to constructive destructive interferences, which gives rise to the contrast in the images that we get from the TEM images that we get. Now, some of the thing is, like, because we are looking at the images from the perspective of the stain, which is essentially like a metal salt, these are less susceptible to radiation damage as biological sample would be. So essentially you have stain, and then in that stain you have these stain excluded regions, which is where your protein was essentially occupying those regions. So you get a difference in scattering. And so essentially the stain per se doesn't get that much radiation damage. So you are essentially preserving as much as possible the ultrastructure of your samples that you're looking at. Then in addition to that, we're kind of basically relying on the size of the stain, the granularity of the stain. So resolution is significantly limited by that. But nonetheless, it gives us a lot of information about how the molecule looks like, what is its shape, and within a range of about anywhere between maximum up to 12 angstrom or 15 angstrom resolution realm, you can get information content of your molecule from these staining methods. So this is one way of preserving the ultrastructure. And the way it is carried out is you have these little grids, these circular grids, on which you have a little thin layer of carbon on which you apply the sample first. And after you apply the sample, then essentially you wick off the excess sample with a blotting paper. Then you put a heavy metal stain like uranyl acetate or uranyl formate, because these are made up of these uranium salts, which are really heavy. They're not radioactive that much anymore, that it has decayed off, but there's a heavy metal toxicity, so we have to be careful when we do these things. And then we wick off the excess stain and then let it dry, and we get a casting of the stain within which our protein is embedded. In the next slide, I'm going to show you. So essentially what happens is this little circle in the middle that you see, that is essentially where your protein or your biological sample would be. And then you have a casting of a stain, which is this black region on top of that. And then as the blue lines are essentially electron waves or electron beam that is essentially hitting the sample. So there are regions which were occupied by the protein, which is very less. I mean, essentially proteins are made up of carbon, hydrogen, nitrogen, oxygen, sulfur. So these are kind of relatively lower molecular weight, atomic weight elements that make up these proteins or biological molecules in general. So they scatter electron much less. But the stain that is made up of heavy metal, there's a lot of scattering that happens. And because of this scattering in the bottom, you have a lot of constructive, destructive interferences, which eventually gives rise to a contrast. And that's what basically the scattering happening from stained area and the stain excluded area, and the interference between these scattered waves is what gives rise to these contrasts. And then that's how you get these kind of a grayscale contrast in your micrograph, the EM micrograph that you see. So you're essentially imaging the stain excluded regions in the case of a negative stain. I hope I'm clear till now. If anybody has any question, again, feel free to interrupt, or I can answer those questions at the end of my presentation. So then here is typically how a negative stained specimen would look like. So there could be regions which are really thick stained. So in bottom, there is just a diagrammatic representation or a cartoon to sort of show. In brown, you have the stain, and then these grayish regions are your molecules sitting on a carbon film. And then you have the entire thing kind of embedded over there. So you see a little bit less contrasty, but these white regions are the regions where there is less scattering. So those are the regions which were occupied by your protein. And the grayish region is where it's all stained by these heavy metal. And that's what gives rise to the contrast in these micrographs that you're seeing over here. On the right side, you have also the same kind of a specimen, which has been negative stained. But you see more contrast over here. But the problem here is we feel that these are like really pretty micrographs and there's a lot of information. But unfortunately, what happens in these cases is part of your protein is sticking out or molecule is sticking outside the stain. So for the regions which are sticking outside the stain, for those you will not get any information. But for the region which is embedded, you get some partial information. So these are some of the drawbacks of this kind of a technique of negative staining that which we use for imaging sometimes. But they're quite, I mean, nonetheless, they're quite handy. And they're easy to do, and they give a lot of information about a molecules, how it looks like and stuff. And if we want to quickly check any of these things, we can do that by negative staining in general. And many of us, what we do is when we have access to such kind of a microscope, and actually in India, many places do have these microscopes where you can do room temperature imaging quickly. So if you look at a Western blot or say a normal protein gel or an SDS-based gel, we just see a band. Now that band essentially tells us that you have a purified protein, but it doesn't tell you any information about the integrity of the protein. So what's cool is this technique takes about 10-15 minutes to do. You can essentially put a little bit of your protein on a grid and just stain it with negative stain and then stick it into a microscope and just collect a few images. And there you can see whether the molecular integrity or the structural integrity of the full protein is maintained or not. Because occasionally what happens is you can have a protein solution which you have purified, but there could be soluble aggregates. The protein could have aggregated. So, and it may not be useful for the purpose for which you intend to use it for. I mean, you may have purified it, but then again, if it's all aggregated, then it's difficult to work with. So in those cases, if you just quickly do a staining also, then you can see, okay, I see my protein over there. This is how it looks like. And it's also kind of very exciting for people like us, where you can actually see how the protein molecule looks like, which no other technique would allow you to do so. By conventional molecular biology techniques. Nonetheless, they are also important. When you purify a protein, you have to check the purity and all these stuff. And then also for a lot of biochemical work, you do need protein gels to be there. But coupled with that, if you have this ability to see how your protein looks like, that takes it to another, I would say, another world or another level of comprehending how things would be and how much integrity is maintained in the protein that you have just purified. So the things are, so these are some of the pros and cons for as any technique would have. Like for negative staining, as I said, it's a very straightforward, quick thing. You get high contrast images. And to a certain extent, many of these uranyl salts also acts as mild cross-linkers to maintain the integrity of the molecule that you're looking at or the protein that you're intended to look at. So the, limitations of this methodology is that the resolution is limited by the granularity of the stain. So if you have, in a case of excellent negative stain sample, you can reach up to maximum 12 angstrom, 15 angstrom. That's kind of the range that you can reach. At that level, you cannot see side chain level or individual amino acid level information, but at least the overall shape of the protein, whether two proteins are interacting with each other. Those kind of things you can quickly check by this thing. And it does give you a 3D volume also from these images, which I'll get to in a moment. And then the other problem is sometimes the protein only adopts limited views, so you don't get all the views of your sample, which is important for getting a three-dimensional map of from those micrographs or from those images. And then because you also dehydrate your sample, because we are imaging dry sample, there's a lot of artifacts that come from your molecules essentially collapsing. It's essentially like taking our grapes and converting it into a raisin. So how it would shrivel. So you cannot get an idea of how the grapes might look like when it was plucked from a grape plant from just looking at the raisins, because it's completely dehydrated. So it's the same thing that can happen over here. And many a times when we are imaging, there's actually no protein. It's just the stained cast that is left behind. So these are some of the sort of drawbacks or limitations, and we cannot get very high resolution information from these TEM images. But as I mentioned earlier, nonetheless, they're very important for giving us a lot of other information as to how the protein may look like, what's its, maybe what kind of symmetry does the protein adopt. And if it is interacting with some partners, you also get information about those things. And it's quite informative and very quickly can be performed. And for several labs and places where there is no access to a Cryo-EM or for high-end microscope, at least this is something which is much more cheaper, and not very cheap, but at least cheaper in some way. And people can quickly have access to such kind of microscopes at places to check some of the samples. And a lot of time, these TEM images are enough for a publication and for papers and stuff. So how do we preserve the integrity as best as we can? So what we do is we freeze the sample on a grid in liquid ethane or propane or some kind of a basically cryogenic liquid. And I'll come to that in a moment as to why we do this kind of different cryogens we use for freezing our sample and not just in liquid nitrogen. So one of the reasons is because we don't want the water or as I said, 80% of us are made up of water. 80% of our body is, made up of water. So even the biological molecules have a lot of water in them. And we don't want them to form crystals, because once it forms crystal, it can actually basically destroy the integrity of the molecule. So that's one of the reasons why we want to preserve the ultrastructure. We quickly freeze it. And then again, once we freeze it, the level of radiation damage also increases. But what's kind of like a disadvantage is that we cannot have very thick sample because they don't freeze quickly. Then we have ice crystal formation. Plus, it also prevents the electron beam from going through. Now, the way we do this is essentially we have these kind of grids or support, and then there is a thin film of carbon or even gold these days, where there are lithographically, there are these holes which are punched over here. You see these tiny holes that are there. And then on top of that, this is like about a 3 millimeter round grid. And then on that, we apply the sample, and then we basically do a lot of sample processing and vitrification, which I'll show in a moment. So we apply somewhat around like 2 to 3 microliter or sometimes even 4 microliter of a sample, but that's way too thick. If we directly take that grid and dunk it in liquid nitrogen or liquid ethane, it's a lot of thickness. It's a lot of volume of liquid that is there. And it's going to be way too thicker for any electron beam to go through, and you won't be able to collect any images in that case. And as I just mentioned, why is it that we can't do the freezing process slowly? We have to use some more other kind of cryogen other than liquid nitrogen. Is essentially because if you slowly freeze water, it forms these crystalline ice. That is the ice that you see in your refrigerator or in your ice machines that are there next to your lab, or even when it snows. So those are crystalline ice, and those crystalline ice are actually hexagonal in nature, and they form when water is cooled down slowly, they essentially are frozen slowly, they form these kind of crystalline ice. And that's actually not good for a biological macromolecule that disrupt the structure, and then eventually your protein is no longer in the form that you want to. Many of you may have used these restriction enzymes and stuff, and you store them in -20 or -80 refrigerators. You'll see very often that they don't freeze. And the reason they don't freeze is because it comes with a lot of glycerol, which prevents it from forming ice or forming any freezing into ice, right? Because that disrupts the structure of these enzymes. And then once the structure gets disrupted, their functionality is gone. So that's one of the reasons why they put a lot of cryoprotectant in that case. If you freeze it somewhat faster, but not very rapidly, then you get these cubic ice, which is in between crystalline and amorphous ice. But then if you freeze it really fast, like extremely rapid freezing happens, then there is no crystalline, the ice doesn't have, or the water essentially doesn't form any kind of crystals. it forms a glassy layer, which is non-crystalline. That's why we call it amorphous ice or glassy or vitreous ice. So this is again an example. So this is all like once an electron beam goes through, you see these diffraction patterns. Like many of you who have done crystallography, you can see these kind of diffraction spots when it's a crystalline ice or hexagonal ice. And then when you get into this cubic ice, which is in between, then you get these ice ring kind of a shape. And then in amorphous ice, which is where we try to image in the case of an electron microscope or cryo-EM, that where water has been kind of rapidly vitrified, you don't get any kind of diffraction pattern over there. So these are all in like Fourier space or in the diffraction pattern space. You don't see these kind of spots over here. It's just completely glassy, and there is no crystallinity left in the solvent. So how do we do it? So this is a case where conventionally this is how it used to be done, and even in my lab here we do the same thing. So essentially this is called as a manual setup for freezing. So you have liquid nitrogen. I'll show in the next image. So there is liquid ethane in this small little cup where you can actually liquefy ethane at liquid nitrogen temperature. And then if I go back to the previous slide, you have a tweezer with a grid and you apply your sample. And after that, you remove the excess sample by literally smacking it with the filter paper, which is kind of heart-wrenching for some of us because it takes a lot of time and effort to purify these proteins. But we smack it with the filter paper essentially to reduce the volume that is left to almost like picoliter, from microliter to almost like picoliter level volumes is what is left on that grid. So it forms a very thin sheet of ice once we freeze it. And that thickness is enough for the electron beam to penetrate through. So if you have all the bulk liquid present on the grid, then it will have a very thick ice, and then that will be impossible to image. So once we do that, we smack it with the filter paper, we remove the excess thing, and then we dunk it into the liquid ethane for rapid freezing, because ethane has very high specific heat capacity, so it can draw off heat from the specimen very fast, leading to its vitrification and not forming any crystalline ice. So nowadays, we also use these kind of fancy robots. I think Kumar Perenbaum, who is going to give a talk at the end of today's session, I think he's going to talk about all these cool toys. I call them toys because they're quite expensive and they're fancy and robotic. We use them also for vitrifying in a more controlled chamber. So this is a vitrification robot called Vitrobot that's also available here at CCMB and many other places, which have a cryo-EM center. So where you use these blotters in a more controlled environment to essentially wick off the excess liquid and then freeze them. So this is an animation from LMB MRC where it shows that you have your protein, you put it on a grid. This is how it would be. You smack it with the filter paper, then it gets dunked into liquid ethane, and then it's all vitrified. And then you have these holes, which I just showed you before. In that hole, you have a thin sheet of ice where your protein is tumbling. Put it in an electron microscope. The electron beam comes through. It goes through a series of lenses, and then you get a micrograph where you can see the zoomed view of your proteins. These are the individual protein molecules that are there. Now from there, somehow recently, we have been able to get like really high resolution structures. Here in the left side, you see these noisy micrographs where you can barely see anything. And on the right-hand side, miraculously, through the processing pipeline, we end up getting like really crystallography level or even better nowadays, structures of large macromolecular complexes. Like how does it happen? Even till date, even though I know how it happens, for me, it's just mind-boggling, and I think it's some sort of a black magic or some sort of a magic that happens. But anyway, I'm going to tell you how this process goes or works kind of as briefly as possible. So you have a vitrified sample, you have all your protein molecules tumbling in this ice, and then you pass electron beam through it, and you get these images, which are essentially projection images. Depending on the orientation in which your molecules are, you get different projection images. Now, these 2D projection images contain all the information content along the axes on which that beam is falling and going through the sample, right? So you get all these information squished from 3D into the 2D based on the orientation of the molecule. And here's a typical micrograph. This is a Cryo-EM molecule, which was classically used in the past for a lot of EM. They were really nice and pretty to look at, and you could easily get some high resolution structures of them. So in this grayish micrograph, you see in this black color thing, these are essentially Cryo-EM tumbling in different orientations. These we call as individual particles. These are individual copies of your protein. Then what we do is we isolate them from the micrographs, we crop it. So before I go into it, it's sort of imagine how the image formation happens. This is from Joachim Frank's book. Like essentially you have a rabbit which is dancing, and then you project light on it and you get a shadow. So here the light is not going through the rabbit, but in this case, in the case of EM, the light, imagine the light or the electron beam in that case is going through the specimen, And that 2D projection image contains all the information content squished into that 2D image on that particular screen, or and in that case, it's a micrograph in case of an electron microscope. So then we isolate these individual particles, we crop them out from these micrographs, and we get a montage of several hundreds of thousands to millions of particles. And then using these computationally, so these are basically, they're classified or sorted and aligned with respect to each other. And then you do sum up all these particles in these different classes, and you get these high contrasty 2D images. These are called 2D class averages, where these have been pixel by pixel, each particle has been compared to each other and then sorted out based on views, or even if there is any variation between these particles, you can sort them out computationally. It's a computationally intensive process where you do a lot of image processing by comparison and stuff, and where we have to go by pixel comparison. And many of us actually use gaming PC equivalent or gaming computer equivalent GPU computers to do all these number crunching, and then finally end up with some more detailed images in 2D. And these are 2D class averages, where each one of these classes contains several hundreds of thousands of particles, which have then been aligned to each other and then summed up across pixel, so that improves the signal to noise and you get more detailed feature from these. And then by combining all these particles, you can end up getting a 3D reconstruction or a 3D map. So essentially what happens is, I'm not going to go into too much details, because that itself is going to take 2-3 lectures to explain the entire data processing or image processing methodologies of EM. You have a 3D object, you get 2D projections or in a micrograph, and then essentially that's the way an image formation happens inside the microscope. And then what happens is you have a series of, so each one of these 2D projections are essentially a slice of a 3D volume. So imagine you have an apple and you slice it into multiple pieces through the center, and then somehow you glue those slices back together, you get back the three-dimensional apple again. So it's something that is similarly happening over here. You get these 2D projection images, you do a Fourier transform of those 2D projection images, and in Fourier space, you get the Fourier images, which each one of those images are basically taken at given angle or different angles of a 3D object. And then once you know the angle, you place those Fourier images together and you get a Fourier volume out of that. And then if you do an inverse Fourier transform from the Fourier volume, then you get back your real object. So that is essentially the image formation in a microscope is happening through the principle of Fourier transformation. And then from those images, you're going back to the real object, that is your specimen. And that's the whole process of data processing that goes on. So somehow magically we do these using different softwares and different algorithms, and we get a 3D volume. And this was the first paper which described this in 1968 from David DeRosier and Aaron Klug. They basically came up with this methodology of getting a 3-dimensional reconstruction from 2-dimensional micrographic images. So this was that classic paper which described this principle, which we still use today to get 3D maps from 2D images of a micrograph. Okay, so then from that process, we get these density maps. This looks very similar to how you would get in X-ray crystallography. From those diffraction patterns, you essentially get back an experimentally derived electron density map. In EM, we don't call them electron density map. We call them electrostatic potential map, because electrons get scattered by both the electrons in the outer shell, as well as electrons in the shells of the orbitals, as well as from the nucleus, because it's charged particles, so that's why it gets scattered by both of them, unlike in the case of X-ray, which is primarily scattered by the electrons. So that's why in X-ray crystallography, we say it's an electron density map. In the case of EM, we call it electrostatic potential map. So in purple is essentially the map that we get, that is from a 3D object. In this case, it's a Cryo-EM. And then at that resolution, you can then, because we know the primary structure of the protein, we can then build in the atomic model by placing, it's like a puzzle where you have to just essentially trace through that density map and build an atomic representation of that experimentally derived map, which is essentially a structure that you typically see in PDB, where all these amino acids are placed and their side chains occupy a certain region of the map, and you build a full 3D map out of your full 3D model of that map that you get from EM. Okay, then most of us then feel, yes, we have got a success, we have a structure now, we can do biochemistry, we can then publish it. So that's typically how we feel like when we get these, okay? So basically, to summarize, what we do is we preserve the sample in near native conditions by freezing them rapidly, excuse me, in kind of liquid cryogen, like liquid ethane. And then most of us then collect these images, but then again, there is radiation damage and certain aberrations that are present in the microscopes, which doesn't let us go. When I started, I said that we can essentially achieve atomic level resolution But unfortunately, because of biological specimen, they're susceptible to radiation damage. There are also other kind of aberrations that happens during imaging, which limits us to get to that atomic resolution. But we are almost near atomic today in terms of achieving the resolution realm from these biological specimens. And then also we rely on the random orientation of the different distribution of particles in that frozen ice. And then sometimes it's limited. We only get limited views. As I said, the three-dimensional map is reconstructed from projection images of the molecule in different orientations in ice. But sometimes we are just really, we are limited by that we don't get all the views. And there are ways by which we can overcome that. Can be a topic of discussion some other day. Some of the good thing is that we get very high resolution information from these methodologies. It's more tedious. So the drawback is, as compared to negative staining, this takes a lot more time and a lot more optimization, almost like crystallography, that it needs a lot of optimization to get good quality micrographs from which you can then recover some of the high resolution information. And then also the contrast is really low because we don't add any contrasting reagents when we do cryo-EM. It's almost in a near native condition that we image it. And then the contrast just comes from the difference in scattering between your buffer and the protein, which is essentially made up of lower molecular weight elements like hydrogen, carbon, nitrogen, sulfur, phosphorus, such kind of things that make us up. Okay, so in 2017, like about 6 years back, the Nobel Prize for recent cryo-EM was actually awarded to 3 people. Jacques Dubochet, who developed this methodology of doing rapid vitrification and maintaining the ultrastructure of biological samples in the vacuum of an electron microscope. Joachim Frank, who came up with the first software called Spider, actually, which helps us basically do all these image processing and get a 3D volume from 2-dimensional projection images of a micrograph. And Richard Henderson, who contributed significantly to several aspects of structural biology, not just in EM, but also more remarkably in electron crystallography, was one of his major contributions, which is why in 2017, this was awarded, when almost recently in the last almost 6-7 years to a decade, when cryo-EM has revolutionized the field of structural biology, not only in allowing us to tackle problems which were otherwise we couldn't have thought about in crystallography because of the sheer size and flexibility and also the complexity. And also we are reaching to a level of resolution where we can easily figure out how these things work.
And education was really good.
Sorry. So essentially, there's also, nowadays we have better cameras. I won't get into much of the details. In the past, we used to image in CMOS or CCD cameras. Nowadays, we have direct electron detector where every single electron hitting the camera can be identified. So we have less loss of resolution or even spread of the resolution. And we get back a lot of the information that is coming on or from the electron waves that are hitting the camera. And I won't get into those details. So this is sort of also revolutionized EM today. And one of the major things from these direct electron detectors are they are fast readout cameras. So if you take an image of something that is happening in motion, you get a blurry image like this. This is essentially a surfer on a wave, and then you can see it's completely blurry. So a thing that happens is when you put these biological specimen into an electron microscope, the moment an electron beam hits, there is essentially a kind of a warping of the ice, which leads to motion in the particles. So these are some virus particles that are imaged. If you would do a regular TEM imaging, you'll see it gets kind of blurred. In cryo-TEM, you'll see these images are kind of blurred, and you cannot get a lot of details from these virus particles other than their general shape. In addition to that, while we are imaging, there are also a little bit of mechanical drifts that happens in the microscope, which can also blur out the information content of the image. But today, because of the fast data readout that is possible, we, instead of taking one full exposure, we take multiple exposures and then computationally align the individual exposures with each other. And that's called as motion correction. And then we can get back these motion corrected, much more crispier images from which we can regain back a lot more information content that what would otherwise would have been possible in the past from CCDs or much more slower cameras. So this is also one of the major reasons why we can get a lot of high resolution information today, primarily because of the camera technology, also with the improvement of the optics of these microscopes. So we can reach to a resolution realm today where we can see almost all the side chain densities, even some water molecules that are kind of bonded or attached, which we can see and reveal them from these EM methodologies. And quickly, I'll just tell you something that we work on is in our lab, we try to understand cytoskeletal elements more importantly into actin cytoskeleton. And there we try to understand these are biopolymers, so how they are nucleated and how those nucleations are regulated. I'll just give you a snapshot of what we could gain in the last 3-4 years from our lab, research from our lab. is essentially we can understand one of these nucleators, which is called as the R23 complex, how it reorganizes itself. It's like a little biomachine. It reorganizes itself from an inactive to an active form, and then that sets the stage for the nucleation of a new actin filament. And this was all done by Cryo-EM. This is just the final product being shown as a comparison from an inactive to an active and what makes it nucleation competent. And then that leads to the formation of an actin filament inside the cell. So this is just a summary of one of those things that we do. And then I can move on to another technique that is there. I won't talk much about it today. We can even do crystallography level electron diffraction, where actually we can diffract a crystal in an electron beam using an electron beam. And then a lot of these small molecules and smaller proteins today can be solved at really high resolution, almost at 1 angstrom level or even close to that. So almost reaching atomic level resolution is possible today. Like you can see, most of these things are 1.5, 1.3 angstrom resolution. These are some of the molecules that have been determined by the structures of which have been determined by micro-ED or microelectron diffraction, which is you use the same kind of a microscope, you stick in a crystal, and that diffracts in the electron beam. And then you get from the diffraction pattern, just like in X-ray crystallography, you can get a structure out of it. There's another thing that we also try to do in our lab, and this is something I started in our postdoc, is try to understand intracellular transport. This is another biological case where EM has been really handy. So if you consider a long neuron, which is over 1 meter in length, and inside those neurons, a lot of transport process happens through certain molecular motors called dynein and kinesin, which work on these microtubule tract. And here you can see these little speckles, which are like little spots or dots. And many of us have seen these in movies, in papers or publications and stuff. But how do these spots look like? What is their ultrastructure? This was not possible for quite some time without the help of EM. And this is something that I did as a postdoc, was to understand the structure. So we used a methodology called as tomography. So many of us know about CT, which is like CT scan, which is actually an X-ray tomography, where a patient sits on a bed, and then there you have a X-ray detector and X-ray generator that goes around in 360-degree space, take multiple images, and computationally combine that together to build a 3D volume of the patient's internal organs and stuff, essentially of the region which has been imaged. It reconstructs a 3D volume, and a radiologist can go through those slices and can see how the organs look like. Of course, the resolution there is in centimeters and meters in scale, because that's actually the dimensions of organs and internal structures that we are looking at. But what we do today is essentially tilt the sample in a microscope. Like this is a tomographic, this is essentially a tilt series where you can see a lot of microtubule tracts. This is frozen in ice, and it's less informative at this point of time, but I'm showing you. But essentially you tilt your specimen, one region of your specimen in an electron beam, and you get a tilt series for the same region. Because you know the angle at which you collected these data, then you can combine them together to get a tomographic volume. These black little spots that you see over here, they're just little nano gold particles. They just help us to align these tilted images with respect to each other. They're called fiducial markers, just like as you would see in the case of essentially in Lord of the Rings. If you would have remembered Smeagol, the actual actor did all the motions and movements. And then there are these little white beads which are then traced to figure out the motion of the animated character, or sort of extrapolate basically the motion of the character from the actual actor. So in a similar fashion, just to trace out like how one tilted image aligns to the other tilted image in a tilt series, you use those gold beads as fiducial markers. So that's why we have those. Otherwise, they're not important as such. And then from that, you get a tomographic reconstruction. That is a 3D volume. I'm just going through on the Z-axis through that volume, and you can see these microtubules crisscrossing with something attached to them in this block of ice that was reconstructed by tomography. And then what we do, just like we isolated 2D images, which I showed you from a micrograph, 2D micrograph, here we isolate volumes, small volumes, which are called subvolumes from this whole tomographic reconstruction. And as you can see over here, these little dynein and dynactin, which are the motor proteins that carry cargo along microtubule tracts, we can see them through some of these Z stack or Z slices from the tomographic reconstruction, and we get these noisy volumes from them. Then we computationally align them to each other and get a 3D volume, which looks like this. And for the first time, we could see how this whole complex looks like. This dynein-dynactin complex actually looks like the cargo carrier inside a cell on a microtubule tract using tomography, which is another methodology that really helps us to view all these bigger complexes. And today, people are doing this inside a cell. Unfortunately, in India, we do not have those instruments to do this inside the cell. But maybe someday in near future, we should be able to procure them. And then we can literally solve structures of these complexes from within the cell, and you don't even have to purify them. by using a combination of tomography and then making thin sections of vitreous samples. So again, the importance is like day by day and year by year, the number of depositions of structures in EM data bank, which is where you deposit your experimentally derived maps, and then also the models associated with it in PDB. So you can see in purple bars, like how many maps are being deposited. This is just from this particular week. And then in the right-hand side, you can see the resolution that we are able to achieve from Cryo-EM today. Like most of the depositions are in 3 to 4 angstrom resolution. And actually, 2 to 3 angstrom is also kind of catching up. It's kind of growing with time. So we are trying to reach that near atomic level resolution, which I spoke about. And someday we should be able to see them. And then in terms of where it's being used, not only academicians like us or who are doing basic research in labs in universities or in research labs in India and other places. Also, companies today are using these for doing drug delivery and drug targets for many of these membrane proteins or many of the targets, where they can design drugs and quickly get a structure to figure out how those are interacting with the protein molecules. And these are some of the publications that I'm showing over here. All of these came from different companies, actually. These are not from academic institutions. Some of them had collaborations with academic institutions. These are primarily all coming from Johnson & Johnson to Pfizer to Genentech, and many of these companies that publish these kind of work in good journals from whatever drug targets they have been able to develop and then check it by structural methodologies. And these are some of the companies that have invested right now in their own cryo-EM centers. And I have several friends of mine who are there working. And these are some of those big biotech companies and pharma companies who are investing in cryo-EM today, because that has essentially enhanced them the capabilities of getting the structure from which people figure out how things are binding and also how it's imparting the function of some of these targets for drug molecules today. And this is from some of the data from our own Cryo-Electron Microscopy Center at CCMB. It's a pan-CSIR facility, but open to any users, academic as well as non-academic users who wants to use it. They can send an email, and then there's a system through which they can also come and collect data of their samples. So these are some of the things that we have collected, and it's almost like 1.9 angstrom resolution we have been able to reach through some of the standard samples. These are some of the other stuff that we work on in our lab, from tomography to other molecules which are of importance. And this is basically my team right now at CCMB. We are also part of ACSIR, with which we have our academic affiliations for the PhD students who come and do PhD. So if any of you are also interested in doing PhD in CCMB, please feel free to check the website of CCMB. Twice a year we have intake through this thing. So feel free to even come here for doing your PhD if you're really excited and interested in doing some research. Otherwise, also people who wants to do project and stuff, a lot of openings come up. And with that, thank you, and I would be happy to take any questions that are there.
Thank you, Dr. Saikat, for that wonderful talk. It gave a very nice overview of the pros, cons, and challenges and the possibilities of Cryo-EM. Thank you. Audience, the session is now open for questions. If you have any questions, you can unmute yourself and ask the question, or you can post it in the chat box. To begin with, Sushant has a question.
Yes, somebody has said motion capture and neuron firing rate. Essentially, one of the things is we get static snapshots from Cryo-EM, just like any other structural methodologies. I mean, we don't get a dynamic sense. What we can get is, these are all like frozen. So if we capture different conformational states from a data set, then we can actually resolve out the different conformations and then tie them in either with existing knowledge from biochemistry, or you can do MD simulations with this structure to get a trajectory as to how one conformation or where it fits with respect to the other. We are all looking at, so we are not looking at dynamic structures. We are only getting static snapshots of dynamic event that may be happening. And from that, we try to determine the dynamicity or the extent of flexibility that is present from those. And we also try to extrapolate from that the level of dynamicity that is happening. So like any other structural methodology, it's all static things that we are looking at. I think Dr. Nagesh has a question you can ask here.
Can I ask Professor Ramakrishnan?
Yeah, please go ahead.
Hi, Saikat. Thanks for a very nice.
Yeah, hi.
This cellular tomography, cryo-ET, is there any size limitation? And for subtomogram averaging, what is the best resolution people have achieved? I have seen 2.5 angstrom. Is there anything better than?
Right now, I think, again, in the 2.5 angstrom resolution realm for something which is in megadalton size, mostly like ribosomes, proteosomes, and things like that, which are really big complexes, primarily because, again, you're limited by the extent of noise that comes from tomographic reconstruction. So anything that is big, we can recover. The signal to noise is much more in that case. So that's one of the reasons why for bigger complexes, people have been able to get by subtomogram averaging, and now the algorithms are also improving in kind of regaining back a lot of the information content and doing much more better picks and stuff. So that's what I would say is the is where it stands right now. But maybe just like in single particle EM, someday we may be able to get even better. It'll be more routine. Yeah.
The smallest size for a single particle, it is 40 somewhere.
Single particle at
for,
least. So for single particle, I can say generally
is the size minimum
40,
size right
40
now.
with symmetry. 40 with symmetry, I would say. Primarily, 40 is kind of an overkill. I mean, sort of like at a limit. So I would say around 60 is where you can basically get really good resolution.
For
60 to 70. Tomography, I think also subtomogram averaging, I would say something around 200 kilodaltons or something close to that would be. But again, it would be in the close to 4 angstrom resolution and stuff that people, 3.54 is where it lies. But mostly for bigger complexes like ribosomes, proteosomes, these have all reached very high resolution right now. That would be my guess. So yeah.
Thank you, Saikat. Thanks.
Welcome.
Audience, are there any other questions? One last question, perhaps.